Scientists from the Helmholtz-Zentrum Dresden-Rossendorf, working together with Wismut GmbH and researchers from the University of Granada in Spain, have made an unexpected discovery involving bacteria and uranium. For the first time, they demonstrated that naturally occurring bacteria can convert uranium dissolved in water into a stable chemical compound when glycerol is available as a food source. Even more surprising, the uranium passed through a chemical state that had previously been known only as a short-lived transition. The findings, published in Nature Communications, could open new possibilities for future research into using bacteria to help clean uranium-contaminated environments. Bacteria living in soil and water already play important roles in ecosystems, and some species are capable of breaking down harmful substances. Previous work by the HZDR research group had shown that certain bacteria can even use dissolved uranium as part of their metabolism when supplied with glycerol, a natural component of plant and animal fats that is also produced during the decomposition of wood by fungi. Building on those earlier findings, the researchers wanted to answer two key questions. How much dissolved uranium can bacteria actually remove from water? And what chemical forms does that uranium become during the process?
To answer those questions, the research team collected mine water from a flooded uranium mine in Germany's Ore Mountains, operated by Wismut GmbH. Inside the laboratory, they recreated the mine's natural oxygen-free environment, similar to the conditions found nearly 2,000 meters below the surface. The scientists then added a controlled amount of glycerol to the water samples, providing the existing bacterial community with a carbon source to support growth. As the bacteria began feeding on the glycerol, they also interacted with the dissolved uranium. After 130 days, the results were remarkable. Only about 5% of the uranium originally dissolved in the water remained. Based on earlier studies, the researchers suspected that the missing uranium had become incorporated into the bacteria themselves. Further analysis confirmed that uranium had indeed accumulated within the bacterial cell walls, demonstrating that the microorganisms had effectively removed most of the dissolved uranium from the water.
The next challenge was determining exactly what chemical form the uranium had taken inside the bacteria. To investigate, the team used advanced microscopic and spectroscopic techniques. Some experiments were carried out at the Rossendorf beamline, operated by HZDR at the European Synchrotron Radiation Facility in Grenoble, France, while additional analyses were performed at the University of Granada. The researchers examined the chemical state, or valency, of the uranium atoms. Uranium is typically found with a valency of 4 or 6, while pentavalent uranium, known as uranium-5, has generally been considered rare and unstable, appearing only briefly during chemical reactions. However, the bacterial samples revealed something completely unexpected. A surprisingly large proportion of the uranium existed in this pentavalent state, suggesting that the bacteria had produced a form of uranium that had previously been observed only as a temporary chemical state. Further analysis showed that the pentavalent uranium had combined with iron and oxygen to form the compound FeUVO4. This compound is relatively new to science and does not yet have an official name. It was first identified in a 2020 study examining soil contaminated by uranium ammunition in parts of Croatia. That earlier research had shown that the compound remained stable for more than 25 years, even after prolonged exposure to atmospheric oxygen. Until now, however, scientists did not understand how the compound formed naturally or that bacteria played a role in its creation. The new study provided the missing piece of the puzzle. Additional experiments reveal that when the dried bacterial biomass was exposed to oxygen, the amount of FeUVO4 actually increased, providing further evidence that the compound remained chemically stable. According to the researchers, this study provides the first direct evidence that bacteria supplied with glycerol as a carbon source can transform toxic uranium dissolved in water into a stable chemical compound.
Although much more research is still needed, the findings raise the possibility that naturally occurring bacteria could eventually contribute to future environmental remediation strategies aimed at reducing uranium contamination. The next stage of the research will focus on understanding these uranium-binding bacteria in greater detail, as well as the biochemical and geochemical processes responsible for forming the stable uranium compound. For now, the discovery offers a new insight into how microorganisms interact with one of the world's most toxic heavy metals and provides a promising direction for future scientific investigation.